Hello, in this video lecture I'm going to talk about the cell membrane. Before we can talk about the cell membrane though, we need to talk about phospholipid bilayers. So phospholipid bilayers are the primary component that makes up the cell membrane. So they determine the structure for the most part of a cell membrane. And of course structure is function, so it's very important that we understand a phospholipid bilayer structure. Phospholipid bilayers also form many of the organelles that we find inside of a cell. For example, the endoplasmic reticulum and the Golgi apparatus and mitochondria, they all have phospholipid bilayers. The fact that the inside of the cell is separated into separate little units called organelles refers back to a concept called compartmentalization, which just means you have all these separate little compartments inside of the cell. And what separates one compartment from another is a phospholipid bilayer. But today we want to concentrate on cell membranes. We're not going to talk about the organelles. So I want to talk a little bit about a phospholipid first. We've learned about these before. Right? So here we see three different visual representations of a phospholipid. Don't forget this is a lipid. But it is a unique lipid in that it has both a hydrophobic and hydrophilic component to it. Okay? Over here we see the structural formula for a phospholipid. This is the space-filling model for a phospholipid, and this is the cute little icon model of a phospholipid. I'd like to draw your attention first to these long components on the phospholipid. If you remember back to your chemistry lesson, these are long chains of carbons and hydrogens. The grays are carbons and the blues are hydrogens. And carbons and hydrogens and long chains are very, very nonpolar. You have lots of carbon-carbon bonds. Those are nonpolar. Lots of carbon-hydrogen bonds. Those are nonpolar, making these tails here very nonpolar. And of course, nonpolar things are hydrophobic. So this end, this bottom end of the phospholipid is very hydrophobic. But if we go up further, we encounter something that's hydrophilic, and it's this phosphate group right here. Let's look at the structural formula for that. See if you can recognize what about the phosphate group makes it hydrophilic. This little line right next to this oxygen, that little straight line does not represent a covalent bond, it's too short. This represents a negative charge. Right? Water likes things with charge or partial charge, and this phosphate group has a full negative charge on it. So water does like this. The partially positive hydrogens on water are going to love that negative oxygen there. So this upper portion of the phospholipid is called the head, and we say the head of the phospholipid is hydrophilic because of that phosphate group. And this bottom portion where we have the hydrocarbon chains, also called fatty acids, that part is nonpolar and therefore hydrophobic. We call those tails, hydrophobic tails. So when in a watery environment, phospholipids will automatically form bilayers. If you were in a lab and you had some pure phospholipid and put it in some pure water, you would automatically form bilayers. In a bilayer, the hydrophobic fatty acid tails face each other. Right, so you have two layers, that's the bi and bilayer. The tails that are hydrophobic face each other, whereas the heads, the circles, face the watery environment. So there's a question on this slide. It says, where is the water in this picture? I didn't write the word water, I didn't draw water, but water's assumed here. See if you can figure out where exactly is the water. Is it going to be here? Is it going to be here? Is it going to be here? Hopefully you guessed that there isn't going to be very much water at all in here. This is very hydrophobic. Water does not like that. Right? The water will be found here facing the hydrophilic heads and over here facing the hydrophilic heads. Phospholipid bilayers are semi-permeable. Some might call this selectively permeable. And they're a barrier, but they're a semi-permeable barrier. What that means is that some things can cross and some things can't. In order to understand why this is, we need to explore further this hydrophobic, hydrophilic nature of the phospholipid bilayer. If we were to measure the width of a phospholipid bilayer, right, we can say maybe it's 10 nanometers. Right? What portion of that is hydrophobic? So how much of that whole 10 nanometer length or width, whatever you want to call it, is hydrophobic? It's maybe about 70%, like 7 nanometers. So roughly about 70% of a phospholipid bilayer is hydrophobic. So most of the phospholipid bilayer is hydrophobic. To understand hydrophobicity is to understand cell membranes. Right? Cell membranes are made of phospholipid bilayers, and to understand cell membrane structure and function, we have to understand the hydrophobic, hydrophilic nature of the phospholipid bilayer. So let's review hydrophobic and hydrophilic. Right? Hydrophobic, water-fearing. Hydrophilic is water-loving. Right? Don't forget, water forms bonds with hydrophilic atoms or hydrophilic molecules. Right? So down here we see water surrounding a single sodium atom. Right? This sodium atom carries charge, right? And water likes things with charge. Over here we see water molecules surrounding glucose. Glucose doesn't carry any charge. I don't see a plus, I don't see a minus. It does not carry any full charge, I should say. But glucose does carry partial charges, right? Glucose has lots of oxygen-hydrogen bonds, which are polar, and that creates partial charge. So water likes glucose. So water forms bonds with charged things or partially charged things. So polar molecules. Are polar molecules hydrophobic or hydrophilic? Well, if you said hydrophilic, you're correct. Like glucose is polar, and water definitely likes that. Charged atoms, like sodium, are those hydrophobic or hydrophilic? And water likes things with charge, so that also is hydrophilic. All right, here I have a list of items, and I want you to try to figure out if these things are things that are going to mix well with other hydrophobic things. So, what mixes well with hydrophobic things are other hydrophobic things. So, if anything on this list is going to mix with something that's hydrophobic, then it itself has to be hydrophobic. So what we have to do is go through this list and figure out which things are hydrophobic and which things are hydrophilic. Anything that's hydrophilic is likely not going to mix with something that's hydrophobic. So let's look at the magnesium ion. Right? We haven't talked about or read about magnesium ions in this class, but we know it's an ion, which means what? It has charge. And if something has charge, does water like it? Yes, anything with charge is hydrophilic. So a magnesium ion is likely not going to mix with anything that's hydrophobic. Phosphate groups. A phosphate group contains a phosphorus and some oxygens, and one or two of the oxygens can carry charge. So if it's charged, it's hydrophilic, and so it's not going to mix with something that's hydrophobic. Cortisol. Cortisol is actually a hormone that's released by the adrenal glands, and it's important in your stress response, and it helps with inflammation and other things. Cortisol is structurally comprised of a steroid, so it's made of a lipid, right? So cortisol is a lipid that's hydrophobic. So cortisol wouldn't mix with hydrophobic things. How about glucose? It doesn't carry full charge. It's not an ion, right? But is it hydrophobic or hydrophilic? Right? C6H12O6. You have a lot of oxygens there. They form oxygen-hydrogen bonds, which are polar, so this will be hydrophilic. Not going to mix with something that's hydrophobic. Hydrogen ions. It's an ion. It has charge. It's hydrophilic. It won't mix with something that's hydrophobic. Water. Well, water does like itself, so water is hydrophilic, right? It's not going to mix with a hydrophobic thing. Amino acids. Amino acids were the subunits to proteins. Amino acids, there was 20 different kinds, but the main components to the amino acid were very polar. You had nitrogen-hydrogen bonds and oxygen-hydrogen bonds there. So for the most part, amino acids are going to be polar and then hence hydrophilic. I will say that there are some amino acids that have a variable group that are hydrophobic. That's something that we don't want to get into. In general, we're going to say that amino acids are hydrophilic. So if they're hydrophilic, they won't mix with something that's hydrophobic. Then we have triglycerides. Triglycerides are a lipid. Since they're a lipid, they are hydrophobic, and they will mix with other hydrophobic things. Okay, so I want to talk about what things can cross a phospholipid bilayer. So we've already established that the majority of the phospholipid bilayer is hydrophobic. Roughly about 70% of the phospholipid bilayer is hydrophobic. So the real question becomes not what can cross a phospholipid bilayer, but what can cross the hydrophobic portion of the phospholipid bilayer. So I have some general rules for crossing phospholipid bilayers unaided. You can cross the phospholipid bilayer if you're hydrophobic, because the phospholipid bilayer is mostly hydrophobic, and hydrophobic things mix well with other hydrophobic things. So if you're hydrophobic, you can cross. Rule number 2, you can cross if you're really small and do not carry full charge. Okay. So small, large, those are relative terms, right? So what do I mean by really small? By really small, I mean 3 atoms big or less, right? So you have to be made of 3 atoms or less. Okay? And you can't carry full charge. Partial charges could be okay, but you can't carry full charge. So let's look at talk about a couple examples, right? What about this little purple triangle? Let's say this is water. According to the rules, can it cross? Well, it's not hydrophobic, right? But let's look at the second rule. You're really small and do not carry full charge. Water is really small. It's 3 atoms, right? And that's the rule. You have to be 3 atoms or less. Does it carry full charge? No, a water molecule does not carry full charge. It carries partial charge. So water actually can cross a phospholipid bilayer unaided, which almost sounds kind of like an oxymoron, right? Because we've established that this area in here does not like water, and water just crossed. Well, the reason why water can cross is because it's so small, it's practically invisible to the hydrophobicity of the layer. And really, water does have a hard time crossing, to be honest, but it can cross. All right, how about this big purple thing? Let's say this is supposed to be a steroid hormone. Can it cross? It's huge. It's definitely bigger than 3 atoms, way bigger. It can cross because it's a steroid, which means it's hydrophobic, so it is able to cross. How about this? Let's say this is glucose, right? Is glucose hydrophobic? Nope, it's hydrophilic, right? Is it less than 3 atoms big? Let's see, it's made of 6 carbons, 12 hydrogens, and 6 oxygens, so no, it's definitely bigger than 3 atoms. And it's polar, and it does carry partial charge, so it's hydrophilic and it's too big, it's not getting across. So you may not cross if you're large and polar, so if you're 4 atoms or bigger and polar, you can't cross. You also can't cross no matter how big or small you are if you're charged. So even if you're a teeny tiny little hydrogen ion, you're made of nothing but a single proton. It doesn't get any smaller than that. It's the smallest thing we really come into contact with in this class is a hydrogen ion, a proton. It is an ion. It cannot cross the phospholipid bilayer unaided. Okay. So let's say this little plus sign here is representing a sodium ion. It is 1 atom big. That's it. It's definitely falling within the realm of you are really small, right? It's 3 atoms or less. It's just 1 atom big. But it does carry full charge, so it cannot cross. All right, what we're looking at here is the same list that we looked at earlier where I asked you, okay, which of these things can mix with something that's hydrophobic? And the whole point of that activity was to come to this activity and see that, okay, the phospholipid bilayer of the cell membrane is going to be mostly hydrophobic. So if you want to cross the phospholipid bilayer, right, you have to be able to cross that hydrophobic barrier, right? So which of the following things can cross the phospholipid bilayer unaided? So apply our rules here, okay? You can cross no matter your size if you're hydrophobic, right? Now, you can also cross if you're 3 atoms or less and do not carry full charge. Let's see who can cross. A magnesium ion, can't cross, has full charge. Phosphate group, can't cross, it has full charge. Cortisol is huge, but it can cross because it does not carry charges of any kind. It is hydrophobic. Glucose cannot cross. It's more than 3 atoms. and it's polar. A hydrogen ion, very tiny, but it cannot cross because it carries full charge. Water, it's 3 atoms or less, so it's small, and it does not carry full charge, so it can cross. Amino acids, they're polar and they're bigger than 3 atoms, they cannot cross. A triglyceride, it's big, excuse me, but it's hydrophobic, and so it can cross. Now, we've been talking about phospholipid bilayers so far as if they were pure phospholipid bilayers, but in reality, there are going to be other things that are a part of this bilayer, like proteins. And the proteins serve lots of different functions, and we'll explore some of this in just a bit. So now I want to spend some time talking about the cell membrane specifically. So we've been talking about phospholipid bilayers in general. I want to specifically talk about the cell membrane. So the cell membrane, also known as the plasma membrane, is a phospholipid bilayer barrier between a cell and its environment. And this cell membrane has many functions. It is used for transport of materials into and out of the cell. You know, it is a barrier. It does keep the outside stuff out, keeps the inside stuff in, but sometimes we need to move things into the cell and move things out of the cell, so we have to get them across the cell membrane. So the cell membrane is used for transport into and out of the cell. The cell membrane is also important for something called a transmembrane potential, which sounds big and fancy, kind of is, but it's really important for how neurons work and muscle cells work, and I'll kind of introduce that concept a little bit later. And another reason why cell membranes are important is because they function in cell-to-cell communication. So this is a picture of a cell membrane. You can see we have our basic phospholipid bilayer, but it's definitely more than just a phospholipid bilayer. The purple things in the pictures are proteins. The blue things, that's actually cholesterol. And the green stuff, those are carbohydrates. So if you notice, the carbohydrates are mostly just found on the extracellular face of the cell membrane, and they're very important in that cell communication thing I mentioned. Lots of proteins in the cell membrane. A lot of them are there for transport, but they have a lot of other purposes too. The cholesterol is kind of an interesting component. The cholesterol helps to maintain the fluid state of the cell membrane. By adding more cholesterol or taking away cholesterol, the membrane becomes more or less fluid. This is a phospholipid bilayer, so it's essentially like a semi-fluid, if you will. And so if we need to make it more solid or more liquid, we can change the amount of cholesterol that's present in order to change that fluidity, if you will. So in terms of structure, most of the cell membrane is made of, hopefully you said phospholipids. Yep, it's mostly made of a phospholipid bilayer. But the cell membrane also contains something to keep the membrane fluid but not too fluid. Hopefully you guessed cholesterol. The cell membrane also contains something on the external surface. It's mostly there for cell identification purposes. Hopefully you guessed carbohydrates. And the cell membrane also contains proteins, which have lots of different functions. So this picture, or these 6 pictures, actually show you some of the functions of the proteins in a cell membrane. Right? And all of the pictures, the blue side is extracellular, and this sort of sandy colored side is intracellular. Right? So the blue is the protein, the gold is the phospholipid bilayer. So here we can see a protein that's allowing circles to come into the cell. So this is a protein that's acting as a transporter. Here we see a protein that is taking in diamonds and turning them into circles, which sounds like magic. This picture is supposed to represent an enzyme. So it's another function of proteins in the cell membrane. Here we see a protein in the cell membrane that's binding to something else, and then apparently some radio signals go off inside the cell. This is representing communication. So this is some sort of signaling molecule that when it binds to this protein, a signal is sent inside the cell. So we would call this protein, by the way, this is a receptor. It's receiving a signal. Here you have a protein with carbohydrates attached, and this is for cell identification purposes. A good example of this is, you've probably heard of the A, B, and O blood groupings. What determines your blood type is actually the presence or absence of certain carbohydrates attached to your cell membrane. Here we see two cell membranes being held together through protein adhesion. So these two proteins are sticking together to hold these cells together. And this is very common because many of your cells in your body, you want them to stay where they're at. And some of them you want to move, like your blood cells, but some of your cells you want them to stay where they're at. You can stick them together using proteins in the cell membrane. And here we see a protein in the cell membrane that's attached to something that looks like licorice inside the cell. This is actually the cytoskeleton. Right? So proteins in the cell membrane are often attached to the cytoskeleton. This is really important in things like your muscles. Okay, so now we can talk about one of the functions of the cell membrane, which is transport across the cell membrane. We need to get things to cross the cell membrane. So what are some of the things that need to cross the cell membrane? Right? So we need to get things into the cell. What sort of things need to get into a cell? What does a cell need to live? Water, food such as glucose, oxygen for the process of aerobic respiration to make ATP. Right, we need to get lots of things into the cell. We need to get wastes out, like carbon dioxide and acids. Something has to cause the molecules to move across the cell membrane. It doesn't happen by magic, right? Energy is what causes things to move. And energy can come from the natural environment, or the cell can use its own energy currency. So there are three main mechanisms that we have to move substances across the cell membrane. If we're not using energy on the part of the cell, that's called passive transport. Energy is being used here, but it's energy in the environment, and the force here is something known as diffusion. That's passive transport. Right? Passive implies, less activity. Right? Then we have active transport. Active transport implies you're supplying a lot of energy. You're very active, right? So active transport is when the cell is actually using its own supply of ATP energy to move something across the cell membrane. And then we have bulk transport. Bulk transport is technically a type of active transport because it does require energy on the part of the cell. But as the name suggests, we're transporting things in bulk, so moving lots of things into and out of the cell. So because it's so different from the other types of transport, we put it in its own category. So I first want to talk about passive transport, but to understand passive transport, we need to understand diffusion. Diffusion is the driving force behind passive transport. Turns out that molecules and atoms are always moving, always vibrating, always wandering around, even in solid objects, even in your laptop or your table or your pen, right? Those solid objects contain moving atoms, moving molecules. This movement can result in diffusion, where molecules spread out evenly into an available space, right? So like, let's say you were holding a helium balloon, right? And you pop the helium balloon. Is it the case that the helium would just stay right there? No, the helium would spread throughout the room that you're in. That's diffusion. We're going to discuss diffusion in terms of a liquid, but it does occur in other states of matter too. So to understand diffusion, I want to remind you about some other terms, like solutes. Solutes are substances dissolved in a solvent. A solvent is the thing that does the dissolving. And when solutes dissolve in a solvent, we end up with a solution. Diffusion is the movement of solutes down their concentration gradient. It's when the solutes move from an area of high concentration to an area of low concentration. So what we have here is essentially a beaker of water, and we've dropped some sort of solute into the beaker of water. Maybe this is sugar, right, glucose. And in the beginning, all of the sugar is right here, but over time, these sugar molecules spread evenly throughout the water. Right? They move from areas of high concentration to areas of low concentration, and that's diffusion. The molecules continue to move until they essentially reach an equal state, if you will, called equilibrium. So what does this have to do with passive transport? Well, during passive transport, the substance being transported moves down its concentration gradient while moving across a cell membrane. And this can occur in two ways. This can occur right across the phospholipid bilayer, where you don't have anything helping the process along, or things can move across the cell membrane by going through a protein that's embedded in the cell membrane. So the names for these processes are simple diffusion and facilitated diffusion, respectively. With simple diffusion, things can move right across the phospholipid bilayer with no help at all, but only special things can do this. So earlier I was talking about what things can cross a phospholipid bilayer unaided. Right? There weren't very many things that can do this, right? You had to be hydrophobic or very small with no full charge. So in a cell, what are the things that can do this? What are things that can simply diffuse? What are things that can cross the phospholipid bilayer unaided? Well, water. Water doesn't have a really easy time doing it, but it can. Okay. Anything that's hydrophobic, like steroid hormones, for example, and triglycerides. Oxygen gas, O2, it's 2 oxygens. It doesn't carry full charge. Carbon dioxide, 3 atoms or less, doesn't carry full charge. And there's a couple other gases. So really you can kind of lump everything that can cross via simple diffusion into 3 categories. Water, hydrophobic, or gases. And that's really it. Anything else has to find a different way to cross. And that different way might be facilitated diffusion. With facilitated diffusion, there's a protein in the cell membrane that's facilitating the transport. It's helping the solute get to the side where there's less of that solute. So this includes channels and carriers. Right? So here on the screen we see a cell membrane. And the pink thing in the middle of the cell membrane is a protein. Right? That triangle I got right across, right? This is an example of simple diffusion there, right? If something moves from an area of high concentration to an area of low concentration, moving from where there's 3 to where there's 1, right? That is diffusion, and it's going right across the phospholipid bilayer, right? That's simple diffusion. Let's see. Ooh, that guy couldn't cross. Maybe that's glucose, right? Glucose is too big and it's polar, can't cross. How about the purple thing? Notice that it went from an area of high concentration down here to an area of low concentration over here. And it can cross big though, so it must be hydrophobic. So this could be like a steroid hormone. That was simple diffusion. Let's look at facilitated diffusion though. So earlier we said that glucose can't cross via simple diffusion. But what if we had a channel or a carrier made out of protein in the cell membrane that could allow for the glucose to cross? Right? Glucose is still following its concentration gradient. It's going from an area of high concentration to an area of low concentration. So that's diffusion. It's just being helped. It's facilitated. So I said there's 2 types of facilitated diffusion: channels, channel-mediated diffusion, and carrier-mediated diffusion. Channels are essentially tunnels, and some of these tunnels have gates. Usually what opens a gate is either a chemical or a change in voltage or a change in mechanics. So we have chemically gated channels, we have electrically gated channels, and mechanically gated channels. And depending upon the stimulus, or I should say that each particular gate is opened by a particular stimulus. Sometimes chemically gated channels are called ligand gated channels, and sometimes electrically gated channels are called voltage gated channels. Some channels only carry specific substances, like maybe only positive ions, for example, or maybe only polar molecules, for example. Examples of channels include sodium ion channels. For example, when we talk about neurons, we're going to be talking about voltage-gated sodium ion channels. With a voltage-gated sodium ion channel, what opens the gate is a voltage change. Right? What goes through the channel, well, if it's a voltage-gated sodium channel, what goes through the channel is sodium. So down here we see some examples of the different types of gated channels. Here's a voltage-gated channel closed and now open. Notice there was a change in voltage as indicated by the change in the plus-minus charge. Here we have a ligand or chemically gated channel. When there is no chemical present, it's closed, but if the chemical comes in and binds, then the channel's open. This is the same thing, just going the opposite direction. And here's a mechanically gated channel. Some sort of physical force, a pressure, movement is what opens this channel. Don't forget, with facilitated diffusion, we're going to use a protein helper. Notice it's still following diffusion, things going from an area of high concentration to an area of low concentration. I wanted to just briefly mention carriers. I'm not going to talk a lot about carriers. So with a carrier, it's very similar to a channel, and they have a protein helping diffusion. With a carrier, there's a little bit more of an intimate relationship between the protein and the substance being transported. So there's some usually like physical touching and pushing and that sort of thing. But I'm not going to go into a lot of detail on that, or any detail on that. Okay, now we're going to look at another type of transport called active transport. So with passive transport, what was driving passive transport was diffusion. With active transport, we're going against diffusion, right? With active transport, we're moving materials against their concentration gradient, right? You don't want the ball to roll down the hill, you want the ball to roll up the hill, right? You're going against physics, right? You're going against the physical environment, and that's going to take energy. Active transport requires energy, right? And this is usually supplied in the form of ATP. Active transport also requires proteins in the cell membrane to help the process. So this is always a carrier-mediated process. There are two main types of active transport. The first type is called active transport. Don't shoot the messenger, I didn't get the name of this stuff. Okay. The second type is called secondary active transport. To make your life easier, we're not going to talk about secondary active transport. We're just going to talk about the first type, primary active transport, which is really just referred to as active transport. The difference between the two, by the way, is how the ATP energy is used. If the ATP energy is used directly, that's the first type. If the ATP energy is used indirectly, that's the secondary active transport. All right, so with primary, if you will, active transport, what we're doing is we're moving things against their concentration gradient. And we're directly using ATP energy in the process. So one example of this is an ion pump. In the picture here, we see a proton pump. This is inside the cell, this is outside the cell. You can see there's lots of protons already outside the cell, right? So according to diffusion, the protons want to come in, right? But protons mean acid. You don't want all that acid inside the cell. So we want to pump more protons out. So you want to move these protons against their concentration gradient. Well, if you want to defy physics, you have to be prepared to spend energy. So we're going to use ATP energy, which we see here in yellow, to pump the protons out. Right? Another example of active transport that's really important is the sodium-potassium exchange pump. So this is the sodium-potassium exchange pump. As the name suggests, it's going to move both sodium and potassium. Okay? It happens to move 3 sodiums out for every 2 potassiums that come in. So the sodium and potassium are moving in opposite directions, hence the word exchange. But they're both moving against their concentration gradient, so this is going to require ATP energy. So you can see there's lots of sodium outside, right? But we're going to put even more sodium outside, so we need ATP for that. The phosphate breaks off of ATP, and the attachment of the phosphate causes a shape change in this carrier. And so now the carrier is open on the outside. So now the sodium is going to be released by the carrier and then will enter into the external environment. Once the sodium is released, it causes another shape change in the carrier that makes the carrier more receptive to potassium. So now potassium can come in and bind. And when the potassium binds, that causes another shape change. Phosphate's released, and now the carrier is open on the inside, and then the potassium will be released to the inside. So basically this protein that is the pump constantly changes shape, allowing us to move sodium and potassium against the concentration gradient. What's helping to change the shape of the protein is the ATP, the attachment of the phosphate group and that sort of thing. So that was the sodium-potassium exchange pump. Sometimes people just call it the sodium-potassium pump. For the most part, this pump is going to be moving sodium out of the cell and potassium into the cell, though there are a few exceptions. The ratio is not 1-to-1. The ratio is 3 sodiums are moving out for every 2 potassiums that are coming in. So we end up with a lot more sodium outside of our cells than in, and a lot more potassium in than out. ATP energy is used to change the shape or conformation of the protein, and the affinity or the attraction of the carrier protein for either sodium or potassium changes when the shape changes. So these sodium-potassium exchange pumps are found in almost every single cell membrane, and there's hundreds and thousands of these sodium-potassium exchange pumps in in just a single cell membrane. And this establishes this status where there's lots of sodium outside of our cells and very little in, and lots of potassium inside of our cells and very little out. And this is going to become important for the function of the cell membrane called transmembrane potential. So students have a really hard time remembering where's the sodium and where's the potassium for the most part. And so hopefully this little picture will help you to remember that. This crazy thing that sort of looks like a purple tree sitting in the bathtub, that's supposed to be a type of cell called a neuron. And if you notice the K's, the potassiums are all inside the neuron, whereas the sodium is outside the neuron, bathing the outside of it. So hopefully that helps you to remember that the sodium is inside and the now I just messed it up. Hopefully that will help you to remember that the sodium is outside the cell and potassium's inside the cell. The last type of transport is bulk transport, also known as vesicular transport, because it uses things called vesicles. Vesicles are little small membrane-bound bubble-like structures inside the cell. There's hundreds of these vesicles inside of our cells. With bulk transport, what we're doing is we're moving things both into and out of the cell in bulk. If we're moving things into the cell, that's called endocytosis. And there's three types of endocytosis: receptor-mediated endocytosis, pinocytosis, and phagocytosis. And then the opposite of endocytosis is exocytosis. So with exocytosis, we're doing bulk transport out of the cell. So let's take a look at the three types of endocytosis. In the top panel here, what we see is the cell membrane is pinching around a bacterial cell. You notice that here, the cell membrane is almost completely pinched around the bacterial cell. Once this part of the cell membrane meets that part of the cell membrane, we get this, a vesicle inside the cell. We call this particular vesicle a phagosome. This process here is called phagocytosis. Phagocytosis is sort of like a cell eating in bulk. Phago means to eat. So we're bringing in larger particles into the cell. And this might be for nutrient purposes, or it could be for like immune system purposes too. In this picture here, we see pinocytosis. Pinocytosis is the equivalent of a cell drinking. Notice that the same process is happening where the cell membrane is pinching around something and bringing it into the cell. But in this case, we're not bringing in large particles, we're bringing in mostly water with small solutes, hence the cell drinking. So that means the bottom picture is receptor-mediated endocytosis. The purple I know my colors, I swear. The blue sort of Y-shaped things are receptor proteins, and upon binding to their target molecule, the process of endocytosis will occur. This is how things like, maybe you've heard of LDL cholesterol. This is how like LDL cholesterol gets into our cells. But I'm not going to spend a lot of time talking about that particular form of endocytosis. So this is a picture of exocytosis, and it pretty much looks just like what I showed you for endocytosis, but in reverse. Here we have a vesicle inside the cell that travels to the cell membrane, and when the vesicle membrane meets the cell membrane, the two fuse together, and then the contents of the vesicle are released outside the cell. So this is how neurotransmitters are released from neurons. This is how some hormones are released. This is how we get rid of some wastes from the cell. This is how we can release some things into our sweat, for example. Now we're going to talk about water transport. Right? So water transport is really important across the cell membrane. Water can cross via bulk transport, but water can also go through channels. We have special channels in the cell membrane called aquaporins, or water pores, and that allows for the facilitated diffusion of water. And then water can also cross via simple diffusion too. So the latter two, in other words, if we're using water channels and simple diffusion, that's collectively called osmosis. And that's what I want to talk about now, is osmosis. So osmosis is the diffusion of water through a semi-permeable membrane. What drives the water movement is solute concentration, oddly enough. Water is following its own concentration gradient, but it appears to be following solutes. Don't forget, in a solution, solutes bind to water molecules. So here, for example, we have salt crystals been dropped into water, and so the individual sodium ions and chloride ions are breaking off the salt crystal because the ionic bonds are weak. And then what's happening is water is forming hydration shells around those solutes, right? The water that's a part of a hydration shell is not free water. It's busy water, if you will. But the water that is not part of a hydration shell, that is free water, right? The more solutes that you have, the less free water you're going to have. So what happens is water moves across the cell membrane to the side where there is less free water. Water is following its own concentration gradient. It's going from an area of high free water concentration to an area of low free water concentration. A lot of students don't like that though, and it's much easier just to say that the water is following the solutes. So here on the screen what I have is a beaker of water, and the beaker of water is separated down the middle by a semi-permeable membrane, such as like a cell membrane. Right? So you can see all the little water molecules all over the place. Right? Now what I've done is I've added a solute to side B. So the smiley face is a solute like urea or glucose or a sodium ion, whatever it might be. Right? So if you notice, these water molecules here are busy and these water molecules here are busy, but all of these water molecules are free. So which side has a lower free water concentration? You said A, you're correct. Let me go back to that slide. So according to osmosis, water is going to move from an area of high free water concentration, over here on A, to an area of low free water concentration. Another way of saying this is that water follows the solutes. Water goes to the sides where there is more solute. All right, let's see if we understand this concept of osmosis. All right, in this scenario, what I have is a cell, shown in red, inside an aqueous solution, shown in blue. Okay. The yellow symbols are representing solutes. So in this case, I have 4 solutes in the solution outside the cell, but only 2 solutes in the cell. Over here, in B, I have the exact opposite situation. I have 4 solutes inside my cell, but only 2 outside the cell. Right? So this is a cell sitting in like a little beaker with water and solutes in it, if you will. So which solution is hypertonic? Remember, hypertonic, hyper means increase, like hyperactivity, right? Hypertonic means increased solute, right? So which solution, not which cell, but which solution is hypertonic? So which solution, A or B, has more solutes? If you said A, you're correct. There are 4 solutes here in this solution, only 2 in this solution. So A has the hypertonic solution. The next question is which cell will experience water gain, right? Well, water moves from an area of low solute concentration to an area of high solute concentration. Water follows the solutes. In A, the water is going to leave the cell and go into the solution because it's following the solutes. So if water leaves the cell is going to shrink. It will crenate. Right? So let's look in B. In B, the water is going to come into the cell because there's more solutes inside the cell. So if water comes into the cell is going to get bigger and gain water weight, right? It'll get bigger. If it keeps gaining water, it will eventually explode. So the answer to number 2 is B. And then number 3 says which cell will crenate or shrink? Well, I think we explained that in A. This cell is going to shrink because the water leaves the cell and comes into the solution because there's more solutes into the solution. So here we have 3 red blood cells, or 3 erythrocytes, and each is in a different type of solution. One's in a hypertonic solution, one's in a hypotonic solution, and one's in an isotonic solution. I'll tell you right now, in panel B, we see a erythrocyte in an isotonic solution. So this is what a red blood cell normally looks like. So which of the 2 red blood cells, A or C, is in the hypertonic solution? Don't forget, if you put a cell in a hypertonic solution, what's going to happen is water is going to leave the cell and go into the solution because there's more solutes in that solution. So if the water leaves the cell will shrink and it will look like this. So this is a crenated red blood cell. The cell in panel A was placed in a hypotonic solution. If you put red blood cells in water, they would eventually get real big and spherical like this and then eventually explode. So this is a red blood cell in a hypotonic solution. The water's coming into the red blood cell because if you put a red blood cell in water, there's no solutes in the water, right? So the water will go from a low solute concentration to a high solute concentration. Water will go into the cell. beaker of water with a semi-permeable membrane down the middle. I just added some solute to side B. So will the water level rise on side A or side B? And don't forget, water follows the solute, so the water is going to go to side B. The last thing to talk about with cell membranes is the specialized function of the cell membrane called transmembrane potential. Transmembrane potentials are also known as just membrane potentials. And these potentials have the potential to do work, and this is due to ion separation across the cell membrane. So it turns out there's a charge or voltage difference between the inside and the outside of a cell. So if you look in the picture here, we'll say the top is extracellular, so outside the cell. And this would be intracellular, inside the cell. And if you notice, I have 5 positive ions outside the cell and 1 positive ion inside the cell. So hopefully you agree that there's a voltage difference between the inside and the outside of that cell. The ions carry charge, and so this charge or voltage difference between the inside and the outside is caused by ion separation. Because there's a difference in the charge between the inside and the outside of the cell membrane, we say that cell membranes are polarized. There's that word polar again. Cell membranes are polarized at rest. Right? Polar like a battery, polar like the earth. It's going to be positive on one side, negative-ish on the other. So most of the cells in your body maintain a transmembrane potential, but this is really important for muscle cells and neurons. There are different types of transmembrane potentials. One type is a resting membrane potential, and that's the transmembrane potential of a cell at rest. Cells are said to be polarized at rest, right, so that polar state exists at rest. The average resting membrane potential is about -70 millivolts. The resting membrane potential is produced, for the most part, by the sodium-potassium exchange pumps. So transmembrane potentials can change, or else they'd be really boring, and I wouldn't be talking about them at all. Right? An ion channel can open, and ions can follow their gradient into or out of the cell, and then we're not going to be -70 anymore. We're going to change. Here I'm letting positive ions in, so I might go to -65. Positive ions came in, so I became more positive. -65 is a more positive number than -70. Changes in transmembrane potential occur in response to a particular stimulus. Something has to open that ion channel, and this is where our gated ion channels come into play. Some sort of movement or a chemical or a voltage will open the gated ion channel. So transmembrane potentials can change by becoming either more or less positive. A depolarization is when the transmembrane potential becomes more positive. If we start at -70, can you give me a number for the transmembrane potential that indicates a depolarization? So we want a number that's more positive than -70. -69 would work, right? -60 would work. -5 would work. 0 would work. +5 would work. All of those are numbers that are more positive than -70. A hyperpolarization is a transmembrane potential that's become more negative, or you could say it's become less positive. So again, if we start at -70, can you give me a number for the transmembrane potential that indicates a hyperpolarization? So if you start at -70 and you hyperpolarize, that means you have to become more negative. So what's a number that's more negative than -70? Maybe you said -80. That would work. Changes in transmembrane potential lead to other changes that are important to physiology. They lead to muscle contraction and your heart beating and your nerve impulses. So these transmembrane potentials are really important.